It starts with a group of cell biologists who were curious enough to find scientific inquiries in mundane questions. What changes occur in the brain when we learn? And once something is learned, how is that information retained in the brain? They devised a reductionist approach that would allow them to investigate the biological mechanisms underlying elementary forms of learning and memory at a cellular and molecular level. This had to be done using an animal that was experimentally tractable, that had a small number of large and accessible nerve cells that could be modified by learning, unlike the human brain. Checking these requirements, the marine mollusk Aplysia made it possible to correlate the observable changes in its overt behavior with the molecular events in the neurons. Now, scientists needed a simple form of learning that could be easily identified and modified in Aplysia. This leads to two simple non associative learning processes. The first, habituation, where there is a decrease in the strength or occurrence of a behavior after repeated exposure to a stimuli. In Aplysia, this manifests in the form of a simple defense reflex, the gill withdrawal reflex, which involves the withdrawal of the gill on the stimulation of the siphon, an action that is like the quick withdrawal of the hand from a hot object. And the second learning process is sensitization, when a strong stimulus brings back either the baseline response or higher to the stimuli after a sudden strong stimuli or stimulation to a different part of the body. They designed controlled behavioral experiments where repeated tactile stimulation to the siphon led to a progressive decrease in gill contraction, indicating habituation. Sensitization was observed when a strong external stimulus temporarily restored the reflex. These findings laid the groundwork for identifying the cellular mechanisms behind learning. Using these raw materials, the scientists built a simple experimental paradigm and an observable circuit from the Aplysia body, which involved the siphon, the sensory component, the interneuron, which connects the sensory neuron to the motor neuron, which in turn is connected to the gill, sending the final signal to withdraw the gill. The story progresses with the scientists eliminating components of the circuit one by one to find the component that is responsible for the habituation behavior to be exhibited. To eliminate the possibility that habituation resulted from changes in the motor neurons or sensory receptors, the researchers used direct intracellular stimulation of motor neurons to bypass sensory input. If habituation were due to motor neuron fatigue, then direct stimulation of motor neurons should have resulted in a weaker response, but it did not. Similarly, they tested whether sensory neuron adaptation was responsible by stimulating the neurons at longer intervals, allowing for full recovery, but the response still habituated with repeated stimulation. This confirmed that the plasticity underlying habituation occurred at the synapse between the sensory and motor neurons rather than in the sensory or motor neurons themselves. They also demonstrated that habituation is a homosynaptic process, meaning that synaptic changes occur only in the specific sensory motor synapse that was repeatedly stimulated. This was confirmed by showing that unstimulated synapses remained unaffected. This was not true for sensitization. Researchers demonstrated that a single strong shock to the tail led to an increase in the strength of synaptic transmission in sensory neurons, which means that unlike habituation, sensitization is a heterosynaptic process. Secondly, they recorded from identified motor neurons and discovered that habituation correlated with a progressive decrease in excitatory postsynaptic potentials, also called EPSPs. Importantly, sensitization restored the EPSP amplitude. The question that followed was the presynaptic terminal of the interneuron the culprit for the synaptic depression during habituation, or was it the postsynaptic terminal where learning took place? This question was tackled in a following paper in 1976, where researchers applied quantal analysis to dissect synaptic depression and habituation. Using electrophysiological techniques, they stimulated sensory neurons and measured the evoked responses in motor neurons given by E. The results showed that habituation was due to a presynaptic decrease in neurotransmitter quanta released, which is given by M, rather than changes in postsynaptic receptor sensitivity, which is given by Q. This was direct evidence for the claim that habituation learning was happening at the presynaptic terminal of the interneuron due to a decrease in neurotransmitter release. To come back to our initial circuit, where we started off by eliminating components, well, it seems that researchers with their starry eyes have found and doubled down on the location where learning happened on a cellular level. To sum up, building on top of simple but clever experiments, scientists found that a simple form of memory, habituation, was mediated centrally, not by sensory and motor neurons, and that habituation was a homosynaptic process while sensitization was not, and habituation was due to a presynaptic change in the amount of neurotransmitter released. If you have stuck around so far, you would wonder which neurotransmitters are those? Well, in the short term, some popular answers are glutamate, serotonin, among many others. For long term habituation, synaptic changes depend on molecular pathways involving gene activation and new protein production. Moving past the cellular level, now we have the molecular answer to learning. If you have stuck around this far, you would think this is a Nobel Prize worth series of work. And you would be right. This is Candle receiving the prize in the year 2000. And that's the story of how a group of scientists built on logical progression and experimental evidence and uncovered the fundamental basis of how and where we learn.